Good afternoon, and thank you for the opportunity to speak today. I am Miriam Mistretta, and I work in Professor Tolosano's lab in Turin. And today I'd like to present my work regarding the dysregulation of heme synthesis and export axis in a new mouse model, FLVCR1A knockout model specific in skeletal muscle. Skeletal muscle is a highly energetically demanding tissue because of the different types of activity it has to sustain. And in this, mitochondria are of crucial importance because they are a very important metabolic hub. Indeed, it is well known that in case of deficient mitochondrial function, we might have muscle pathologies. Moreover, mitochondria are also very important for heme metabolism, as the majority of heme biosynthesis occurs in mitochondria, and it also requires some intermediates that are in common with important metabolic pathways, such as the TCA cycle. And heme is a fundamental molecule for life because it is a cofactor of enzyme, and it is also the prosthetic group of hemoproteins. It is synthesized in every cell, and its content must be tightly regulated at different levels, one of them being the heme synthesis and export axis through FLVCR1A. And FLVCR1A is a heme exporter located on the plasma membrane and ubiquitously expressed. It is important because it controls heme export, and thus it removes the intracellular heme content in excess that otherwise would inhibit ALS1, which is the first and most important enzyme in heme biosynthesis. Thus, by exporting heme, FLVCR1A sustains heme biosynthesis. And skeletal muscle is one of the major heme consumers because it needs heme to incorporate it into myoglobin. And indeed, heme synthesis and heme metabolism has been already studied by many groups in skeletal muscle. But no one has ever investigated the coordinated heme synthesis and export axis and the role of FLVCR1A, despite also being demonstrated that FLVCR1A is also a regulator of energetic metabolism. Therefore, we have decided to generate this new mouse model. First of all, we have assessed the efficiency of deletion in skeletal muscle, and then we have collected some information about progeny and body measurements. These mice are vital, they are prolific, and at least at the macroscopic level, they do not show any difference compared to control mice in terms of total body weight and single muscle weight. However, if we look more deeply into morphology, we can see some differences. Indeed, especially in 10-month-old mice, we can see a great reduction in cross-sectional area. And at this point, we wanted also to investigate the fiber types as well, because we know that usually type 1 fibers are the smallest one. So we analyzed fiber types, but we did not see any difference. So FLVCR1A deletion actually affects muscle morphology without causing an alteration in myosin heavy chain isoform expression. At this point, we moved to the analysis of metabolism, and we performed some metabolic assays on tissue cryosections. As you can see, the SDH activity and complex 4 activity are reduced in our knockout mice, and these results are not due to an intrinsic incapability of these enzymes to work, because we have also isolated these enzymes. We have performed the same metabolic assays in vitro by providing all of the needed substrate, and these enzymes seem to work properly. So it is possible that in vivo we have a situation in which we either lack some substrate or there are some morphological impairments that prevent metabolism from working properly. Of course, we have thought about oxygen as the lacking substrate, and we have injected an hypoxia probe, and we have seen that there are some hypoxic region very specific in some fibers only in our knockout mouse. We think that this might be due to a defect in oxygen storage, because we have also observed decreased myoglobin, and this is in accordance with other results that we have obtained in our laboratory, showing that by interfering with heme synthesis and export axis, we also interfere in hemoprotein synthesis, because there is not enough heme to be incorporated in them. So we are rather confident that this sort of local hypoxia that we are seeing is due to a defective oxygen storage, because we have also analyzed vasculature, but we did not observe any difference. So these mice are equally perfused compared to controls. On the other hand, we have also observed some morphological changes at the mitochondrial level. These are very preliminary electron microscopy images, and as you can see, mitochondria are very unusual looking with the rarified crests, and they are rich in these electrodense materials. So we have also caused a sort of damage in mitochondria. And overall, these morphological and metabolic defect that we are observing also impact on muscle function, because our knockout mice perform worse compared to control in every type of test that we have performed, from strength to endurance and coordination. And this is particularly true in older mice as well. So in conclusion, if we have functioning heme synthesis and metabolism, we have functional mitochondria and effective metabolism with no change in morphology and muscle function. Whereas if we compromise heme synthesis and export axis, we cause a change and a defect in morphology and in mitochondrial metabolism, which also has an impact on muscle function, which is poor. So at this point, even though we are not before an overt skeletal muscle pathology, we think that these results are interesting, and we want to investigate them more, because we think that this model might be useful both for basic biology research and also to identify new targeting approaches for metabolic diseases in skeletal muscle. A special thanks to Professor Emanuela Tolosano and Veronica Fiorito, who are with me today, and thank you all for the attention.
I was impressed by your SDH and COX staining, which you have in the central fibers, completely absence of staining.
Yeah.
Is it just because there are no mitochondria, or because the mitochondria are compromised? And the second point is related to the signaling pathway. So heme can control, there is a kinase that can sense heme, can control protein synthesis. So have you checked if in your mice protein synthesis is compromised or is blocked?
Okay, for the first question, this is very difficult, because the reason why we have decided to perform electron microscopy was just this, because we have seen this sort of void inside the fibers, and this is not due to thaw and freeze cycles, because in other type of staining we did not see it. So we actually did not explain this. We cannot understand if it is because of the mitochondria that are non-functional or if it is because of the lack of myoglobin, for instance. So we are now investigating deeply this aspect. Regarding the second question, no, unfortunately, we haven't checked, but maybe this is an important point to investigate as well. Thank you.